Questions, you know the importance of that from first lab practical. Most of the questions are going to be just like that, except for the real tubes and stains. Okay, so from this particular lab, you need to know the pH formula, which is the first one over there. You need to know what is the meaning of halophilic and halotolerant. Then, yeah, that's enough from that part. Okay. And I don't think nobody is going to ask pH range and stuff, even though I asked for lab quiz. Nobody asks the pH range, but you can expect the question normal pH of bacteria. That is somewhere 7 plus or minus 0.2 or 6.8 to 7.2. That makes sense to you when I said 7 plus or minus 0.2?
Mm-hmm.
Yeah, it can be anywhere from 6.8 to 7.2. pH range will not be there usually. In case if it is as you know the answer, it's 4 to 9. One is pH range, one is optimal pH. That's the difference between two questions. Is that clear, all of you?
Mm-hmm.
Then you need to know halophilic, halotolerant. After that, UV. The same question I asked for the lab quiz. It's the same question from your lab practical question. Okay, so the problem caused by UV, how it can be repaired. It's actually elaborated answer. We are expecting, like you have to say problem is timing timer or thymine will come together, create a bond. As a result, polymerase enzyme cannot move, so no replication properly. Corrected using photolyase enzyme, which is activated with the presence of light, photoreactivation. Same answer I can write. After that, what you have to study is if tubes are kept over there, you know to find out halophilic one, right? Look for the maximum growth based on question. Remember, first practical you got based on temperature. So this time it will be based on pH. It will be given like pH 7, pH 3, pH 2. Optimal pH for that organism, you have to look for maximum turbidity. Otherwise, question is, this is a acidophile. You have to pick which tube is acidophile. How you will find that? Look for pH, whichever below 7. Acidophile means look for the lowest pH. If you have pH 2, you should pick that one. If you have pH 2, pH 6.8, pH 8.5, you have to pick pH 2 because that's the lowest of the pH. So based on the question, you have to pick A, B, C, 2. That's the question expected from first section. Then we are going for oxygen requirement. A lot of expected questions are there. First thing, you have to identify gas bag. What is it used for? Gas bag is used for? Come on, we are here for review. Gas bag is used for?
Aero.
What? Anaerobe.
Anaerobe.
Strict anaerobe. Candle jar is used for?
Facultative.
Facultative.
Plus
anaerobe.
want CO2.
So if both are kept there, you are asking for a comparison. How you will write?
One is facultative.
One is for strict anaerobe, one is for facultative anaerobe. Okay. Now, what is the purpose of candle in a candle jar?
Candle.
It's two.
Candle, fire requires oxygen.
So
So
it
whatever
burns.
oxygen present inside
It'll get
that
rid of it.
candle will utilize for flaming. Okay, that's the purpose of candle. So once it is over, fire over, gone means that means all oxygen has been utilized. But doesn't mean that oxygen will go inside. It will little bit, not like all amount. Okay. Then gas bag, when you study, you need to know what is the purpose of methylene strip, what is the purpose of that pouch, what is the content of that pouch, function, or what is the purpose, why you using that. Now you need to know, if you have a tube, you have to say the thioglycolate growth enhance the growth of what type of bacteria. If it is on top, it will be strict aerobe. If it is bottom, strict anaerobe. If it is like a little bit down on top, microaerophile, all over facultative anaerobe, otherwise aerotolerant. So this you need to know. That will be from the real tube. So make sure not to shake the tube, just lift it up just like your last practical. Okay? Lift it up, see, keep it down. Otherwise, you bend yourself down and see the reaction. Another thing is, what is thioglycolate, where you have used that? You have to say what is thioglycolate. It is used for oxygen requirement test. Next question is, you will get plates like this. You will be asked to write whether what type of organism is bacterium A based on oxygen requirement. So if you see growth everywhere, what you will write? Facultative anaerobe, very good. If you see growth only in gas bag, whereas you don't see any growth in ambient and candle jar means strict anaerobe. If you see growth in candle jar and ambient air but no gas bag means cannot say aerob since candle jar is there. Okay. So based on how you see the growth, you have to find answer. That is another possible question. Then we went to unknown identification. So unknown identification, make sure to study this chart right from gram staining. So first, a biochemical test always you do for an unknown identification is
Gram
staining. Then we did lot of tests. You have to know everything from this chart, like a positive reaction, negative reaction, if reagent used, what is the reagent, if it is indicator, what is the indicator. After API, sometimes there will be a strip. You should be able to identify that strip. That is API test strip. Now you know what is advantage, what is disadvantage, isn't it? Either it will be one advantage, one disadvantage, or two advantages and two disadvantages, but it's going to be there. Now urine culture. When you talk about urine culture, you need to know EMB. What is the purpose of EMB? How a growth will be seen in EMB if it is a gram-negative bacterium. Usually positive indicate the presence of a metallic green sheen. Another thing from EMB is lactose fermentation. So make sure to study the lactose fermentation part and this metallic green sheen part. Now, ENB inhibits the growth of this type of bacteria. ENB inhibits the growth of, it was in your lab quiz. ENB inhibits the growth of gram positive or gram negative? Gram positive. It will enhance the growth of gram negative. So make sure to read the question carefully, then answer. EMB growth. Blindly, please don't write answer. Check whether it is enhancing or inhibiting. Okay. Another one is CNA. Then inhibitor in CNA is. What is the inhibitory agent in CNA? Somebody wrote NA last time for lab quiz. Please don't write like that. You have to write it as antibiotic naladixic acid or simply naladixic acid. If you just write NA, it's not going to help, okay? That you have to know. So CNA we will usually use for the growth of gram positive. It will inhibit gram negative, just opposite to EMB. Then we went to staph and strep interpretation. So common question is one similarity, one difference between staph and strep. Then we have used SM110 and DNAs only for staph or strep? Staph. That's actually used only for staph. I saw that question once, like SM110 and DNAs was used for the test of which type of bacteria, which genus of bacteria. So you should answer as staph. Then another thing, MSA. That's a very common question. If you see growth on MSA with the yellow colonies present means you have to mention about both. First thing, it is salt loving or halophilic. Second thing, to utilize the sugar mannitol. Whenever sugar is utilized, acid will be produced. Whenever acid is produced, phenol red indicator will change color from red to yellow. It's the same thing for carbohydrate utilization also. Then you have to know serological testing. It was today's lab quiz. Make sure the antigen used in monospot, dipstick. Dipstick is used for strep infection, especially strep thought. Staphyloslide is a confirmatory test for Staph aureus or S. aureus. Whenever you use staphyloslide, I asked a question for today's lab quiz. Are you checking for antibody produced against an antigen or an antigen itself? What is the answer for that?
Antigen.
Antigen itself, because we are checking for Staphylococcus bacteria, isn't it? Staphylococcus bacteria is considered as an antigen itself. I'll usually see that, very frequently I'll see that question. So that's why I put the same question for your lab quiz. Now I'm going to show you some slides. I don't know how far you can answer it, but make sure to go through motility test also this time, because we have done that for biochemical test. So in motility, you will not get the depression slide and stuffs like that, but you will get the TTC medium. So you need to know what is TTC, where you have used that. Then all of you know negative and positive of motility test, right? If it is straight down, one thin line, negative. If it is spreading, then it is positive. What is the purpose of Durham tube in some of the biochemical tests?
Gas production.
Gas collection. So based on which medium you are getting, you have to answer that. If it is carbohydrate utilization medium, you have to answer to collect carbon dioxide. If it is is nitrate reduction, you have to say it is for collecting nitrogen gas. Yeah, based on whichever medium you are getting. So how you will interpret the result using a Durham tube?
If there's a bubble.
Empty space or a big bubble. What all are the 4 main tests you are checking using IMBIC? Indol, methyl red, phosphorus, citrate. These are the 4. Don't mention about motility or hydrogen sulfide, okay? We cannot write that if they are asking for 4 main tests. Now, what we are checking for in Bose-Poskett test? Otherwise, what is the difference between methyl red test and Bose-Poskett test? Both we are checking for glucose utilization, isn't it? But what is the difference? VP is, yeah, very good. VP, end product will be acetone. We are checking for acetone. Whereas methyl red, we are just checking for glucose. What is the reagent used for VP test? VP test, Barrett Very good, Barrett. When you come to citrate test, citrate, make sure whether there is a royal blue on top. Don't expect it will be like whole half tube will be royal blue. Some of you got slight on top, right? Whenever you see royal blue, you have to consider it as positive. Okay. Why we are using a wooden stick in oxidase test?
Because it can react with the metal of the loop.
Huh?
It reacts.
Because it will react with metal. We will get a false result. What we are checking for in catalase test? No, we are not checking for bubbling. What we are checking for in what is the purpose of catalase test? We are checking the presence of exoenzyme catalase. Reaction is it will convert hydrogen peroxide to water and oxygen. Oxygen is what you're seeing there as bubbling. Nitrate reduction, you should be very careful. Immediately when you get red color after adding reagent, then you got nitrite. Those who are not familiar with molecular formula, NO2, NO3, please don't write that, okay? The starting product is, the starting stuff is nitrate or NO3. It is reduced to nitrite, otherwise NO2. If you are not getting reaction after adding reagent, then you will add zinc. Okay. So question will be like before adding zinc or after adding zinc. Before adding zinc, you are getting a tube with red color. What will be your answer?
It's positive.
Positive for what? If you write positive, it's wrong. Positive for nitrite. Excellent. After adding zinc, you are getting red color. What will be your answer?
Positive for nitrate? No. No, it's negative for everything.
No reaction. That's what you are supposed to write. Don't write negative for everything. No reaction. Whereas, if you are not seeing any color after adding zinc, what will be your interpretation?
But you had a false test on the first one because there's some positive somewhere.
Positive for what?
Nitrate.
Ammonia. Positive for ammonia. You should be very careful when you answer that particular question. Where is nitrate reduction? See here, it's clearly given in this table, tabular column, okay? If nitrate is reduced to nitrite, you will get red color after adding reagent. But after adding zinc, if you get red color, pink to red color, that means no reaction. If there is no color at all, that means nitrate has been reduced to ammonia. If you don't know NH3 is a molecular formula, please make sure to write ammonia because NH4 is ammonium. Some of you will write NH. I saw NH4 sometime. That's ammonium. It's an ion. Okay? Just write it if you are not familiar with the molecular formula.
What's the purpose of that?
Zinc will check whether ammonia is produced or not. That's the purpose of zinc. Okay. Now, what are the two major end product of nitrate reduction? Ammonia and nitrite. Some of you for your lab quiz wrote nitrogen gas. I gave you point that time, but now I am correcting it. If you are studying from your lab quiz, please don't study that. Major end products are nitrite or ammonia. Then, what is the purpose of zinc powder? We discussed already, right? When you will add it? When you will add zinc powder?
You don't get a reaction.
You don't get a reaction after adding nitrate reagent. That's the time you will add zinc powder. Okay. What do you mean by exoenzyme? Secreted outside of the cell.
Cell.
That is exoenzyme. Give any one example. Anything you can write, catalase or whatever you have done here is actually exoenzyme. Amylase, lipase. I don't have a color printout, or else would have been very helpful for you. Okay, now when you come to starch, all other enzymes are very much related with their substrate, like lipid, lipase, hydrogen peroxide, catalase. The only difference one is amylase. So amylase is used for what type of hydrolysis test?
Carbohydrates.
Starch. That's the only different one from the substrate. So if amylase is as, you should write it as starch hydrolysis. Otherwise, the enzyme perform starch hydrolysis reaction, that is amylase. So that's the only different one. Otherwise, just compare it with the substrate name and then write your enzyme name. What is the name of fat hydrolysis enzyme?
Lipase.
Fat hydrolysis enzyme.
Lipase.
Lipase. Lipase is fat hydrolysis. Sometimes they will not as lipid, it will be asked as fat. But you should answer it as lipase. Where you have used Kovac reagent? Test. Questions can be like any twisting way. You never expect it will be like straight question. Sometimes they will just keep. Oh, another thing. Grams iodine, if it is kept over there, you'll be asked where you have used this reagent other than gram reaction. What will be your answer?
What was that?
Where you have used grams iodine reagent other than grams reaction.
That was in the purple.
Starch.
Yeah.
Very good. Starch hydrolysis. Okay. Now D-aminase and D-carboxylase. First we will discuss about D-carboxylase. D-carboxylase. D means removal. So carboxylic acid group is removed from, otherwise COOH group is removed from amino acid. So your end product will be alkaline or acidic. When acid group is removed, end product will be alkaline. Okay. In D-aminase, amino group is removed. Remember, amino acid means one side carboxylic acid, one side amino group. Otherwise, one side acid, one side base. When D-aminase is happening means amino group is removed. Now what is left in that amino acid? Carboxylic acid, isn't it? As a result, this will be acidic. Keep this concept in your mind, then right answer. So you did only one D-aminase test. What was that? Just one D-aminase test. Phenylalanine. You just check with one amino acid, phenylalanine, D-aminase. Whereas carboxylic acid we did with multiple. We did with arginine, lysine, or methane. Purple color will be positive, yellow color will be negative. Whereas in phenylalanine D-aminase, positive will be
Yellow.
Is it
No.
green or the
avocado
blue?
green.
Green.
It's avocado green. That's positive. Yellow is negative for D-carboxylic acid test. That arginine, ornithine, and all those things. Okay. What are the three reactions you are checking when you use SIM medium? Indoor H2S, otherwise sulfide, and motility. Very good. How you will check whether your organism is producing H2S or not? By checking black color. Motility, looking the effusion or spreading, but it can give you false result. So what you will do, even if you see a motility in SIM medium?
TTC.
Excellent. You will perform TTC and do the confirmatory test. Okay, yeah, that's excellent answer. So whenever acidic pH is formed, what will be the color? Yellow. Yellow is acidic pH and alkaline will be the other one. Okay. Alkaline, we are actually starting with purple. Okay, so now next one is gelatin hydrolysis. It's just opposite to coagulation. In gelatin, if you see liquid, that's positive. Solid, negative. Coagulation is just opposite, which was the confirmatory test for our last one. There, if positive, has to be solid. Negative means it will be liquid. So study that. Gelatin, we are checking for the presence of exoenzyme dash.
Gelatinase.
Gelatinase. What is the purpose of keeping your tube on or in ice during gelatinase test or gelatin hydrolysis test?
To check for solidification. No.
To bring it back down to room
To
temperature.
bring the temperature to the normal one, because we are taking from incubator, which is like high temperature 37 degrees Celsius. Usually your tube or your medium will be liquid, so you're bringing it temperature back to normal. Otherwise we can keep in room temperature for 10-15 minutes till that hot temperature has gone or that warm temperature has gone. Now when you talk about EMB, the speciality of EMB is both the differential media and selective media. So you need to know what is a differential media and what is selective media. Here we are checking for selective media by selecting gram-negative, and differential media by checking the utilization of
Mannitol.
lactose, not mannitol, lactose. What are the two dyes present in EMB media? Or indicator?
What is what now? I'm sorry.
Two dyes, otherwise indicator present in EMB media.
Eosin and
Very good. It's the name of the media as such. Eosin methylene blue. That's why we got the name EMB. Those who like to go through this will give the complete explanation of what reaction happens, why it happens. Okay, just go through if you want to. If you don't want to go in detail, fine, leave it. I'm not forcing you to go through
what
this.
is this?
Hmm?
This is a
No, it's my personal note. Those things you don't have here, just and all those things. Okay. Take. Let me ask you a few questions. I don't know how many of you prepared for your test. This is from your lab practical review online, along with your lab manual. I told you, one of my students gave this to me long back. Okay, so where you have used this 3% H2O2. Suppose if you are getting a bottle like this, which test you have used this? Catalase, excellent. It is a reagent for catalase test. H2O2 stands for hydrogen peroxide. Excellent. You got a tube like this. Okay, this is tube A. This is tube B. Tell the reaction in tube A and tube B. Otherwise, compare the reactions in tube A and tube B.
Two is negative.
This is a phenol red sugar probe, or you are checking for carbohydrate utilization. Tell me. First, talk about tube A.
negative.
Positive for carbohydrate utilization. That's why you got the yellow color as it is produced. But what about gas? Negative for gas. Whereas tube B,
Positive for gas.
positive for gas, positive for acid. So you should say specifically positive for acid and positive for gas. No matter whether they ask question like that, sometimes interpret your reaction. That's all will be there. You should mention specifically about gas and acid. Is that clear? Just like today I asked you one, if you bile esculent, you have to mention about both bile resistant and esculent utilization. So interpret means whatever available results are there, you need to write everything. This is a MSA medium, okay? MSA plate. What about this result and this result? Yellow will be positive for mannitol utilization plus resistant to salt. If you are getting growth, that means it's obvious that it's resistant to salt. See, so nowhere it's mentioned. They are just talking about mannitol. But when you write answer, we expect you to write both salt resistant and mannitol utilization. Is that clear? Suppose you are getting a plate like this. You are seeing a growth but no yellow color. What will be your answer?
Salt resistant.
Excellent. Salt resistant but cannot utilize mannitol. So this is also an example for both selective medium and differential medium. Are you getting the difference between selective and differential? Differential will differentiate whether it is utilizing that or not. Whereas selective means it will just select. For example, organisms which are haloduric or halotolerant can only grow in MSA medium. Like gram-negative organism can only grow in CMB medium. That is known as selective. Differential means it will compare two type of species. This is oxidase test, positive, negative. Sometimes you will see pink color. That's what you see during our lab time, right? So pink is also positive. Okay. What is the reagent or indicator used in oxidase reagent? I mean, oxidase test. Oh, you have a wonderful name for that. Remember? The easy name is oxidase reagent. 2,3 tetradiamide. It's a long name. Anyone of you remember that? It's okay. If you just write oxidase reagent, also I'll give you a point. Okay. Just to console Tony. Tony is eating my brain like anything, saying that hard. See, he's staring from there. My dear, that was wrong. I cannot help you in that.
I'm just listening. I'm not thinking about that.
Oh, okay. Thank you. Thank you. I mean, if 0 is coming on right hand side, it has a lot of value. Keep that in mind. Now, this is catalase test. So if you see bubbling reaction, that is catalase positive. If this test is there, you have to perform yourself. Remember last time you did the same thing, isn't it? No, you did something by yourself. Which one was that? I asked you to put it in bleach bowl once you are done. I remember. It was this side of the table. Which test was that? Something you were performing yourself and you were putting the whole slide in bleach bowl? No?
It was the last slide. It was too large.
Okay, then maybe I'm thinking something else. Anyhow, if this particular test is there, you will be given culture. Hydrogen peroxide will be there, slide will be there. You have to take the culture, make sure to add hydrogen peroxide, see the bubbling, and you're supposed to put everything in a bleach bowl. So it will be a self-performing test. This is the only thing you'll be performing during your lab practical. All other things will be kept over there. Okay, so this is gelatin. So if you are getting gelatin, make sure to take the tube, tilt a little bit, check whether it is liquid or not. Again, whichever tube you are taking during your lab practical, make sure not to hold to the cap. Hold the tube as such. Don't break it. There will be a penalty if you break. That means you are going to get 0 for your lab practical. You are spoiling the future of all other students followed by you. So don't do anything like that. Be careful.
So you said the liquid will be positive?
Positive for gelatin. Okay? Coagulase, it's opposite. Now, this is a skim milk agar. So, a skim milk agar, casein. Very good, casein. We are checking for the presence of enzyme. Caseinase. Don't expect question will be very straight like that. So, a medium will be kept there. Skim milk agar will be written. Question will be what you are checking here. You have to say you are checking for the presence of enzyme caseinase, which will break down casein. This is decarboxylase test, where you can see positive here. You know that yellow color means it's positive. Here it is shown as purple, but reality, what color is this?
Fusion.
It's a fusion color? Okay, whatever it is. Reality, we saw it as purple, right? Deep purple, like violet purple. As indole test, positive means you will see on top that pink color. Okay, on top. Usually it will be pink color and it has to be undisturbed. Anytime if indole test is there, make sure not to take the tube. Just look like that. What is this? API test strip. Otherwise, API 20E. Why we are saying that 20E? We can do 20 tests at a time. And what is that E stands for? Huh? Excellent. Enterobacteriaceae. That's why API 20E. You should answer the whole stuff. API 20E, the strip. Okay. Now, all of you know if positives, negatives given means you can find. All of you are sure how to find the 7-digit number, right? I saw somebody was carrying that. All of you know that? You will be having positives, negative, add all positives together. What value you get, you have to fill the first bubble. Like that, you are supposed to find 7-digit code. In case if whole negative is there, your answer is 0. But make sure to write 0 there and find a count whether you have 7-digit or not. If it is 6-digit, it's wrong.
Did you say the last digit on the right hand side is always negative?
Oxidase will be always negative because Enterobacteriaceae, it's an oxidase negative bacteria genes. Okay, so no matter whether it is given or not, you are expected to know that oxidase will be negative, will not have any credit value there. So I'm again telling you, in case if you get a 0 digit, make sure to write that 0 there. Don't think 0 means I can skip it. You need to have 7-digit code at the end. Now, this is nitrate. I think we already discussed. Before adding zinc or after adding zinc reaction. Then second question is, what is the purpose of zinc? Then you will add it. What is this? Excellent. Staphyloslide test. It's a confirmatory test for
Staphylococcus aureus.
Staph aureus. How you will confirm a positive reaction?
Agglutination.
Agglutination. What is the purpose of blue color?
Just let
Blue
it be
latex?
show easy.
Just to make the visibility, or make it visible or easy visibility. I don't know what is this, by the way. Let me read the question. Oxygen requirement. Oh, thioglycolate probe. That I already told you, right? If it is all the way on top, aerobic. All the way bottom, anaerobic. Little bit down the top, microaerophile. You see all the way from top to bottom, facultative and good. Bile esculin. So bile esculin, again, it's a two test. One, we are checking whether organism is resistant to bile. Second one, utilizing esculin. So if you see a growth, that means resistant to bile. If you see black coffee color, utilizing esculin sugar. Good. For which organism we have performed bile esculin test? Staph or strep? Huh?
It's for strep.
Straight.
Staph or strep?
Strep.
It was for strep. Whereas SM110 was for
Staph.
Staph is catalase positive or negative?
Positive.
Strep is catalase positive or negative?
Negative.
Staph and strep are gram positive or negative?
Positive.
Huh?
Positive.
Oil of glucose you don't have. Okay, this is EMB. See that metallic green sheen color, which is lactose positive. Whereas if you are not getting any color like that, it can be either purple or metallic green sheen. Both will tell that it is lactose positive. Whereas if you are just seeing growth over there, no color, that means lactose negative but gram negative. Is that clear? Any amount of growth on EMB medium is gram negative. Purple color, metallic green sheen, lactose positive. Just growth without any color, lactose negative, but gram negative. Okay? That means EMB, why you are staring me? Is that clear for you? Any amount of growth on EMB plate will tell that it's a gram negative organism because it will support only the growth of gram negative. Second thing is there is lactose inside. If lactose is utilized, you will see either purple color or green metallic sheen color. So to be on the safer side, if this question is asked, tell that lactose positive organism will give purple to green metallic sheen color. So your part is safe. Okay? If you just get a growth, no purple, no green metallic sheen, what is your interpretation? It cannot utilize lactose, but it is gram negative. Does that make sense to you? Okay. This is your, what is this? Yeah, same thing. Same thing, but there is no metallic green sheen. It's just showing purple color, lactose positive. So lactose negative means you will just see the growth, no color. Now, I'm taking you back to the old last lab. All of you know how to pick a good streak plate, right? All of you. Look for the maximum single colonies, that will be the good streak plate. Second one, whenever you are doing any biochemical test, you will go for, I know someone of you did that mistake when you were doing biochemical test, but I was keeping on telling you, whenever you do biochemical test, don't do zigzag. Just do a straight to and fro streak. Why?
To observe the pattern.
To observe, not pattern, to observe the result very clearly. Remember last class somebody showed me a zigzag and asked me how I will measure the zone of inhibitions. You cannot do that if it is like that, you know. So in order to see the result very clearly, usually you will not go for zigzag, you will go straight up and down for biochemical test. For example, you are checking for amylase test. Imagine if it is zigzag, how difficult to see the result. Whereas if it is straight, it's very easy to see the zone. Okay, this is urea. Urea positive will be pink color, deep pink color. Yeah. What is the reagent used there or indicator? can study from the tabular column. This is phenylalanine deaminase. This is DNase. Yeah, DNase is another thing I want to discuss about. You have two types of DNase medium. One is with the reagent, one is with the indicator. So question can be asked like anything. What is the reagent used for DNase medium means what will be your answer? FCS. Okay. What is the indicator used for DNase medium? Methylene, methyl green. Sometimes they can ask you what is the exact function of methyl green in DNase medium. Don't write indicator, you have to tell what exactly happening there. Likewise, what is the exact function of HCl in DNase medium? So prepare for that also. Now starch hydrolysis. So starch hydrolysis is the one we are using grams iodine other than gram reaction. What enzyme we are checking here? Amylase. What is this? You haven't used something like this. So obviously I will keep that box. This is actually a gas pack. Okay, this is a picture of the old gas pack we had here. Right now we replaced it with the box. So that will be kept over there. You need to know very specifically purpose of methylene blue strip and purpose of that pouch. Okay. Interpret your reaction for nitrate test seeing this too. After adding nitrate reagent. Interpret your reaction for nitrate reduction after adding nitrate reagent. Before adding zinc.
Gas positive for
Gas positive for the
Gas
first
positive,
tube.
but
Okay.
it's negative.
And
It's
negative for
Nitrite.
nitrite. Excellent. Second tube.
Negative for both.
Negative for nitrite. Negative for gas. Good. Point B, G you haven't done. Okay. You have tube number A, tube number B. Which is positive for coagulation test? A or B?
B.
B, isn't it? You are checking it with rabbit plasma.
Rabbit plasma. No.
We haven't done it. Litmus milk you don't have. Okay, what is this?
That's a
Dip
dip
strip,
strip.
otherwise strip for strep.
Strip,
Okay.
strip,
This is a test usually do for strep throat infection. Who was giving that? Joe or Randy? Somebody was talking about this today morning. Who was giving that presentation? Randy? So all of you know what is a positive reaction, what is a negative reaction, right? 2 red line positive, 1 line negative. This is methyl red test. Motility similar. We are very familiar with that now. Yeah, we are done. See the positive of citrate. Sometimes you will get like this, all blue. Sometimes only on top you will see blue. Wherever you see a blue, no matter whether it is all the tube or a little bit, write it as positive for citrate. Okay. So you have filled your biochemical tabular column, all of you. Make sure to do that before you go. That's very important. Otherwise you will have a tough time. You want? Sure. Anyone of you need another one?
Yes.